Good evening, everyone. Welcome to another free session on Botany Insider. So, we have the third session for you today. This is general at the GATE XL Biochemistry segment, right? Just check whether the audio and video is clear to you or not, and then we'll begin with the class, right? So, daily base के ऊपर हम previous questions को revise कर रहे हैं. We are learning how to practice out and how to go forward in the entire preparation. You have your examination in a month, so every single day we are trying to solve as many questions as possible. Good evening everyone. Good evening Aditi, good evening Jangya. Okay, let's begin. Right, so just a quick reminder, we have our rankers batch started today. If you wish to enroll in, you can enroll via downloading our application, right? It's a course for your CSIR UGC net life sciences preparation. In addition, we have our exam cracker modules available. If you're someone who is preparing for GATE examination dedicatedly, so you can take up these GATE revision capsules. They are going to help you out in the best manner. We have a mock test series also, which will help you in solving out the questions, solving out as many questions as possible for your upcoming exam, because एग्जाम के अंदर जितनी ज़्यादा हम प्रैक्टिस करते हैं उतना बेटर रिजल्ट आता है, राइट? एंड वी हैव आवर डिटेल्ड ई बुक्स ऑल्सो अवेलेबल, राइट? ओके, let's begin. You can also join in our Lakshya batch. This is a completely free of cost segment for you all, in which every day a task is given to you, a free test is conducted, so that students can practice out and have a good guidance for their preparation, right? So yesterday we discussed all these questions already. We have to, I think, start from, there was a match the following question. Let's just quickly check that. Which was it? Aditi, I gave the homework as well, right? That question was given as homework. I think this was the question. Yes, I think this was the question that was given as homework. We discussed this question as the last one. Raghav, we do not have any free book kind of a thing. If you wish, you can come in the Lakshya batch. Over there, every day a small PDF is shared in. You can take up that, but I would suggest you can come up in the revision capsule segment as well. It's a last minute revision thing, very very cost effective. Not, we are not charging very heavy price for this. It's available in a very nominal pricing. And right that we discussed, or this was the homework that I gave you in the previous class, right? Mark the answer for the question, everyone. You have column 1 and column 2. In column 1 you have solubility, you have ion charge, you have polarity, and you have the molecular size given. And over here you have different types of segments, right? For example, technique of chromatography, centrifugation, salting out, isoelectric focusing, gel electrophoresis, and everything. So mark the answer for this particular question, then we'll do it together. Yes, mark the answer for the question, everyone, then we'll solve it out. I hope you all have done this homework. So this is the revision capsule that I'm talking about. If you wish, you can have this. Yes, mark the answer quickly. So over here you have solubility, over here you have ionic charge, over here you have polarity, and over here you have the molecular size. So what is the format, or which different biological separation methods are used in different segments? Mark the answer. Okay, done. Okay, let's see. So answer for this is option number C. Solubility is linked to salting out. Ionic charge, isoelectric focusing. Ionic charge can very easily be correlated with the isoelectric focusing. Polarity is reverse phase chromatography, and then molecular size is ultracentrifugation. What is the concept of centrifugation all about? Centrifugation is nothing but it's a process in which we have the movement, or we have a very fast movement, or a very high speed movement done in, right, in the rotor, if you can recall all of those things. So with the help of, or based upon the molecular size differences, we have the like separation done in. So what should be the answer? This is molecular size is ultracentrifugation, polarity is isoelectric focusing, that is R with 4. Where is R with 4? No, R polarity is, sorry, polarity ionic charge is isoelectric focusing. Q with 4, S with 2. What else? Solubility is what? Salting out. P with 3. What else? Polarity. This is with your reverse. Correct. So C is the answer for the question. Everyone participate in the questions. Okay, next question for you all. Which one or more of the following statement is or are correct regarding the electromotive force generated by the electron transport chain? So when we learn about the electron transfer chain, which of the statement is or are correct? That means it can have more than one statement also correct in. That means it is a multiple select question, right? Try the question, everyone. It is used for synthesis of ATP. It is not used for active transport process. It includes pH gradient, and it does not include an electrical potential gradient component. Mark the answer. Done. Very simple. Which of the following statement is correct in terms of, or is or are one or more than one statement correct in terms of generation of electromotive force that is generated by your electron transfer chain? Everyone participate in the question. No, it is used for synthesis of ATP. It is not used for active transport. It includes a pH gradient component, and it does not include an electrical potential gradient component. A and C. Okay. Others also mark it. Let's see. Very good. A and C. We need to find what is correct in terms of the same. So it is used for ATP synthesis, and it includes a pH gradient. Not used for active transport process is wrong, and does not involve electrical potential gradient is also wrong. It is used to make ATP, and it is composed of your difference in the pH concentration, right, and the membrane potential. As a result, B and D, both of these are false. Next question. This one is related to the transport and retention of protein. Again, a multiple select question for you all. Question is in terms of your transport and retention of proteins in the different cell organelles. First option is mannose 6-phosphate residue is involved in targeting proteins to lysosomes. Transport of protein into the mitochondrial compartment is aided by positively charged amino acid residues at the N terminus and internal hydrophobic segments. Retention of proteins in the ER lumen requires KDEL sequence motif, and then at the C terminus, then nuclear protein are transported in an unfolded conformation, and nuclear localization signal is subsequently cleaved by the peptidase in the nucleoplasm. We have to find out which information is or are correct in this term. Mark it, then we'll do it together. Mark the answer quickly. Let's see which of the following statement is or are correct regarding the transport and the retention of the proteins in the different cell organelles. So, is the mannose for 6-phosphate required for your protein targeting to lysosome? Yes, it is required. Then transport of protein into the mitochondrial compartment is aided by positively charged amino acids at the N terminal. Maybe. Then if I want to retain any protein in the ER, it would require KDEL sequence. May be correct. Then last option, nuclear proteins are transported in an unfolded conformation, and the nuclear localization signal sequence is subsequently cleaved by the peptidase in the nucleoplasm. So last option is wrong because your nuclear localization signal is not cleaved off when, or only the peptide signal, the signal sequence which is required for the transportation, that gets cleaved off. We do not have the removal of the nuclear localization signal. First statement is correct, second is correct, third is correct, right? Three statements are correct. So this is a multiple select question. If the question would have been which of the following is incorrect, then the answer would have been option number D. So what is the correct answer for this particular question? The answer for this question is A, B and C, right? All three statements are correct, right? I hope it's clear. Yes. Okay, let's proceed to the next question. Okay, next question about the fluorescence spectroscopy. Again, which of the following statement describe or describes the fluorescence spectroscopy? So this question is again a multiple select question. So you have to give it a try to this question also. Everyone mark this, then we'll do it together. Try it out. Okay. Mark the answer quickly for this question. Which of the following statement correctly describes fluorescence spectroscopy? Emission maxima is independent of the excitation wavelength. Emission maxima depends upon the concentration of the quencher. Emission maxima varies with the solvent polarity, and the emission maxima varies with the temperature. What should be the answer? Which of the following describes fluorescence spectroscopy? So A, C, D, all the three things are correct. It does not depend upon the quencher's concentration. Except for this, all the other things are correct. That is, it is independent upon the of the excitation energy, right? It varies with the solvent polarity, and your emission maxima varies with the temperature as well, but it does not depend upon the concentration of the quencher. So ACDR correct. Another question about the processing of your pre-mRNA, right? Which statement is correct, or which statements are correct in the processing of your pre-mRNA in the eukaryote? Try this. Try the question, everyone, then we'll do it together. Yes. Mark it. Which of the following statement is or are correct in the processing of mRNA, pre-mRNA in the eukaryotes? Okay. Let's see. 3 dash to 5 dash exonuclease activity is involved in the conversion of pre-mRNA to mRNA. 5 dash capping and addition of the 3 dash poly A tail precedes the splicing. Splicing of pre-mRNA occurs via trans-esterification reaction, and then alternative splicing can yield different mRNA products from the same pre-mRNA. So what should be the correct answer for this particular question? C and D are the correct statements. Splicing is carried out by two trans-esterification reaction, and yes, alternative splicing will be able to generate different formats or different products as a result of your pre-mRNA splicing happening in. Then the 3 dash to 5 dash exonuclease activity is not required, because 5, like, what is the functionality of 3 dash to 5 dash exonuclease activity? The exonuclease activity is for your proofreading, so this is not required in the splicing part, right? And then polyadenylation does not necessarily precede splicing. That is also correct, because it's not a compulsion for splicing to occur only after the capping and tailing has been completed. If you have learnt about the pre-mRNA, mRNA, like the entire central dogma, you should be knowing about it. Very basic question, right? A and B are wrong, C and D are the correct statements, right? Hope it's clear. Next question about the addition of puromycin. What will be the changes when I have the addition of puromycin done during the eukaryotic translation? So during the eukaryotic translation, if I'm adding one of the inhibitor, for example puromycin, what changes will you see over there? It resembles the amino acyl end of the charged tRNA. It occupies A site, P site, E site. Yes, proofreading functionality is of exonuclease activity that is not required at all during the splicing part. It is majorly required when we are having the DNA replication occurring in specifically, right? Mark the answer for this now. Good evening, Reet. Mark the answer for this question. Okay. So, what altered segment, or what do we get to see? What changes do we see upon the addition of puromycin during the eukaryotic translation? So, puromycin resembles amino acyl end of the tRNA, charged tRNA, puromycin occupying the P site, E site, A site. What should be the answer? A and B. It occupies your A site and it resembles your amino acyl end of the charged tRNA, right? It does not mimics your P or E site at all. So very simple, very what do I call, factual question altogether, right? Hope it's clear. Next question. Okay, let's try. I think you can do this question, or people usually don't end up doing these questions. We'll keep on doing only the theoretical part for the time being. Okay, let's try this. This one is an easy question. When cell components are fractionated by sedimentation, so if I am using the technique of sedimentation, so with the respect to the gravitational force occurring or the gravitational force acting upon different organelles, what is the level of uh, or which component gets separated, or what is the order of the components getting separated? Lower to higher gravitational force. In the gravitational force is denoted by, or it is calculated with what? It is calculated with G. The unit is G. So we have to find out the correct order from lower to higher gravitational force. So let's see how many of you can mark the answer for this now. Give it a try to the question, then we'll do it together. Mark the answer. From lower to higher gravitational force, which component gets separated first, which component gets separated afterwards, from lower to higher order? Okay, let's see. So very good, A is the correct answer. First you have the nuclei, then you have the mitochondria, then you have microsomes, and then you have the ribosome. So in the case of a differential centrifugation, larger organelles, like larger or denser organelles, they pellet at lower G, and the smaller one require progressively higher gravitational force. So first will be nuclei, then you have mitochondria, then you have microsomes, and in the end you have the ribosomes availability. So A is the answer for the question. Easy and simple, right? Another question. This is again very easy. So In a population, the probability of a susceptible individual getting infected with SARS-CoV-2 is low when a majority of individuals in the population become immune. So what is this phenomenon known as? This is a very common segment or very common term we all heard about during the COVID time. So try to mark the answer, then we'll do it together. Very simple. So question is, if a person has not taken is having the or what is the probability of this person getting infected if the area where he lives, let's say there are 100 people and 90 of them already have gotten immune to the virus. So what is the probability that this particular individual will be having lesser chances, right? So how do we get to solve this particular thing? Yes, it is called as herd immunity. That is an immunity which occurs because of the population, right? So this is herd immunity. So when most of the people are immune, the transmission chain breaks, which leads to lowering of the infection rate altogether, right? When we were learning about, or when we all were in the state of COVID timing, so over at that time also government tried to focus upon herd, the concept of herd immunity, that is trying to make the maximum people immune so that the chain of COVID, the chain of that particular virus getting transmitted reduces, and as a result chances or the probability of someone getting the infection even if the person has not taken the vaccine reduce, right? So that is what, sorry, herd immunity is. Next question. Now you're given four different pathways, four different steps, I would say, right? Question is asking you that given below are four of the reactions of a glycolytic pathway that is catalyzed by enzyme E1, E2, E3 and E4. So first one is fructose 6 phosphate E1 and converted into fructose 1,6 bisphosphate. Then you have fructose 1,6 bisphosphate converting into dihydroacetone phosphate and glyceraldehyde 3 phosphate, which is with E2. 3 phosphoglycerate with E3 converting into 2 phosphoglycerate, and then phosphoenol pyruvate with E4 converting into pyruvate. Question is asking which of the enzyme E1, E2, E3, E4 is or are not a part of gluconeogenesis pathway. Try the question. Which of the following is or are not a part of gluconeogenesis pathway? Mark the answer for this. Then we'll do it together. I'm doing fine, Reet. How are you? Yes. Mark the answer. Very good, Aditi. Why aren't you marking the answer of all the questions? Not learnt everything till now? And if you people want to revise all these things in one go, we have revision capsules available for you. I am pinning up this particular link. So if anyone who's interested, you can purchase and it is going to help you out a lot. So given below are four of the reactions. We need to find out which of the following is or are not a part of gluconeogenesis pathway. So A and D, first and fourth, are not involved in the gluconeogenesis pathway, right? Second and third are involved in. So first and fourth are not involved in. Keep a note of this. That's so good to know. We have so many other lectures also. Make sure that you watch them. Next question. Which of the following molecule is or are second messengers produced in the phosphodiinositide signaling pathway? Which of the following is or are second messengers produced by phosphodiinositide signaling pathway? Mark the answer. Easy. Again, it's a multiple select question. You might have two options as the correct answer. Mark it. Phosphatidyl inositol 4,5 bisphosphate. Mark it. Which of the following molecule is or are second messenger produced by phosphoinositide signaling pathway? So, what should be the answer? Very good, very good. Let's see. B and D is the correct answer. Yes. Phosphatidyl inositol 4,5 bisphosphate. Now this is talking about the entire IP3 DAG pathway that we have, right? So phosphatidyl inositol 4,5 bisphosphate, this is the substrate, not a secondary messenger. IP3 is a secondary messenger. IP3, it is not biological relevant, like over here that we are, whatever they are talking about, over here it is not relevant, because what you have to check the positioning of the phosphate 145 or 135. There was a question asked in CSIR also once based upon just this pointer. It was a 4 marker and the question was based upon this same segment of where do we have the presence of that phosphate group being there. So it is not 1 3 5, it is 1 4 and 5. Then DAG, it remains in the membrane and it activates your PKC. So which of the following are the secondary messengers? So IP3 and DAG, these two are the secondary messengers and not phosphatidyl 4,5 bisphosphate, because PIP2 is the one that does not carries any message. It is the one which helps in the breakdown, or it is the one which will be like participating as a substrate and not as a messenger, right? PIP2 gets converted into IP3 and DAG, if you can recall the entire pathway over there. Hope it's clear. Next question, let's do another. Okay, try this. Which of the following amino acid has more than 2 acid base groups? Mark the answer. Which of the following amino acid has more than 2 acid base groups? Very good. Mark it. Mark the answer. Which of the following amino acid has more than 2 acid base groups? Alanine, leucine, phenylalanine and tyrosine. So for this particular question, you should be able to recall that molecular structure of the various amino acids. Let's see. Answer is tyrosine. Very good, very good. So there's only one amino acid which has more than two, or more than two acid base groups attached to it, right? Another question. Okay, let's try this one. This one is group 1 and group 2. uh, Group 1 has a number of different immune cells, and over here you have a number of related concepts. So antibody production, first cell to be recruited at the site of infection, antibody dependent cell mediated cytotoxicity, and histamine production. So try the question, everyone. Mark the answer. Sahil is late to the class. Class timing is 4. You said you would be joining daily. The cell types with the associated processes that we have. So NK cells, natural killer cells, B cell, mast cell and neutrophils. So D is the answer for this particular, C is the answer for this particular question, right? So NK cells, they are antibody dependent cell mediated cytotoxicity. Then you have B cells, antibody production, mast cells, histamine production and neutrophils. These are the first cells to be recruited at the site of infection. Till 5 we have the class. 4 to 5 daily. We started off with this series yesterday itself. Four statements about lipids are given below. We need to find out which of the statement is or are correct. You can see the questions of GATE examination are not difficult as compared to CSIR. But the point is, you have to have a different approach towards these questions, right? Because over here you have multiple select questions as well, you have different types of other type of questions as well, we have NAT type of questions as well. So make sure that you are focusing upon almost the same theoretical part, but the format of approaching these questions is a bit different. Okay, mark it. Mark this. Yes, first statement is that cholesterol is amphipathic. Second is self-assembly of phospholipid in the water is due to the hydrophobic effect. The temperature at which the gel phase changes to liquid crystalline phase increases with the increase in the degree of unsaturation of fatty acyl tails. Choline head group lipid is positively charged. Let's see. Four statements about the lipid are given below. We need to find which statement is or are correct. You have A, B, and D being the correct one. Cholesterol is amphipathic. Yes, it is. Self-assembly of phospholipids in the water is due to hydrophobic effect. Yes. Phase transition temperature increase with unsaturation. No, it reduces the packing. So TM decreases. Last is choline head group is positively charged. Correct. So what should be the answer? A, B, and D. C is the wrong statement. Again, this is a multiple select question that we talk about. Another question: which of the following techniques can be used to separate the proteins according to their molecular weight from a mixture of protein? So I have to separate my proteins based upon the molecular weight. So which technique am I going to use? That's what the question is. Ion exchange, size exclusion, sodium dodecyl sulfate, and that is SDS page, and you have sucrose density gradient centrifugation. Mark it. Okay, just one question. How many of you are preparing for ICMR exam? Or how many of you are preparing for the BRET examination? Specific name. Some people get confused. So how many of you are preparing for the examination which is on 10th of January? Which is by the Department of Health from. Yes, how many of you are preparing for DHR BRET examination? Anyone in the class right now? Okay, so I have curated a free test series, like 5 test series for you all. If you people want, you can join in. I am sharing if this message can be transferred here. Okay. Okay. No, I'm not able to send in all of the tests. So I'm not able to send the entire link over here. Let me see if I can. Okay, so these are the free mock test that we have. This is for, no, it's not going, it is not getting sent. Yes, you can give that as well. Obviously, no worries in that. I don't know why the message is not getting transferred. Okay, so this one is the microbiology one. Um, this is biological sciences. These are free tests. Okay. This is biological sciences. So if you're interested, you can give the test. I also have curated one for part A. Like in the examination, you have part A, part B, part C. So one for part A, one for part B, and for part C, there are different subjects. So I've curated it for 3 subjects right now. One is biological science, one is microbiology, and the other one is biochemistry. So this one is biochemistry. Okay, so if anyone is interested, you can give up these free tests as well. It is going to help you. We have a lot of free tests for the GATE examination also. Okay, every day we are having a free test for GATE XL examination. You can join there also. Okay, let's come back to the question. The question is, which of the following technique can be used to separate the proteins according to their molecular weight for a mixture of various proteins? So the first one is ion exchange chromatography. Second one is uh, size exclusion. Third is sodium or SDS page specific, and last is your sucrose density gradient centrifugation. So answer is B and C, right? These two are there which can be used for the separation based upon the molecular weight, right? So B and C. Ion exchange chromatography separates based upon the charge. Sucrose density separates based upon the density or the sedimentation, right? Not the molecular weight. So B and C is the correct one. Let's do one more question for you all. Okay, let's try out this one. Which of the following immune cell listed below are agranular? Mast cell, monocytes, T cells, and eosinophils. Mark the answer for this. Yes, we have a daily test. We have daily tests. Aditi can tell because she is also there in the Lakshya batch. So she can tell you better. She has been there in the class in that course since long, I think a month or so, if I'm not wrong. So there are like almost 900 students in that particular free guidance batch. So this is the free guidance batch that we have. Mark the answer for this. Which of the following immune cell listed below are agranular? Let's see. So R and S, that is your monocytes and lymphocytes are agranular, except for this, your eosinophils and your mast cells are granule rich cells. So R and S is the correct one. C is the correct answer. Another question: which of the following enzyme is located in the outer mitochondrial membrane? Which of the following enzyme is located in the outer mitochondrial membrane? Citrate synthase, fumarase, monoamine oxidase, and succinate dehydrogenase. Try this. Mark the answer. Which of the following do we have, which is located in the, which of the following enzyme that's given to you is located on the outer mitochondrial membrane? Citrate synthase, fumarase, monoamine oxidase, and succinate dehydrogenase. So the monoamine oxidase is the one that is located in the outer one, right? So it is to the outer one. Then you have citrate synthase, fumarase, matrix, and succinate dehydrogenase is in the inner membrane. So for this particular type of question, you need to remember the segment of where the part is, or the entire structure of the same, right? Another question: which of the following statements about the DNA polymerase 3 of E. coli is not correct? So if I'm learning about my DNA polymerase 3 in the E. coli, which of the following is the not correct statement? Try this. Which of the following statement about the DNA polymerase 3 of E. coli is not correct? So it catalyzes your Nick translation. Its absence is lethal to E. coli. It synthesizes a complementary DNA strand using a single stranded template, and it possesses a 3' to 5' exonuclease activity. Answer for this is option number 1, right? That is, it catalyzes the Nick translation. This is not the correct statement. Nick translation is what? It is the functionality of DNA polymerase 1 and not 3. So polymerase 3 is majorly required for what it is required to add on the segments to the single stranded DNA template. right? And it has 3 dash to 5 dash proofreading activity. So answer for this is option number 1. Last question for you today. Which of the following compound is not a translation inhibitor? Which of the following compound is not a translation inhibitor? Chloramphenicol, cycloheximide, puromycin, and rifamycin. Mark this, last question. Which of the following compound is not a translation inhibitor? Try it. Which of the following compounds is not a translation inhibitor? Chloramphenicol, cycloheximide, puromycin, and rifamycin. So what should be the answer? So rifamycin is the one that inhibits your bacterial RNA polymerase, right? So chloramphenicol, cycloheximide, and puromycin inhibits translation, but all the other one are the one that inhibits the bacterial RNA polymerase, that is transcription. So from here you can analyze and check, see to what level a question has been asked. That is, all of them are inhibitors, but you have to find, like, keep a note that which one is inhibiting the translation and which one is inhibiting the transcription altogether. So you can see the fact, or the your command over factual statement is very, very important, right? So what should be the answer for the question? The answer for this is option number D, right? So I hope till here all the questions are clear to you and you have understood the questions till here. I'm giving you one question as homework. Take this particular question. Those of you who are there in the class, you can take a screenshot as well. Take this question as homework and make sure that you remember to solve it for tomorrow's session. Tomorrow again 4 p.m. we'll have a session. Be there in the class. We'll discuss another set of 10 to 15 questions. For 1 week we'll go for this biochemistry. I'll take up botany as well, I'll take up zoology as well, I'll take up microbiology as well. So if you people join in on regular basis and frequently, I will be taking up the sessions till your February examination, but you have to make sure that you're participating in and coming in the classes on daily basis. Okay, so that's about it. Let's meet in tomorrow's session, 4 p.m. Anyone is interested in joining up any of the sessions, you can join. CSIR's rankers batch is ongoing. We started off with the theoretical part today in the morning. So you can join in. There's a free 2 classes available. You can watch them as well. Let me share the free batch thing as well. Wait a second. So if you wish, you can join in these free segment. You're most welcome, Reet. You're most welcome. Most welcome, Aditi. So this is the free class for June 2026. If you wish, you can join in. Then we have our GATE XL capsule course also available. Like there's a flat 30% off available for you all till 8th, so make sure that you join in before that. You have a mock test series for GATE XL. We have GATE XL's notes also, no, like full detailed notes as well. For GATE BT, your biotechnology segment, I am curating a revision capsule for you all for the GATE BT as well. Just give me I think 1 or 2 more days, you'll be getting this GATE BT capsule as well, because many students are demanding about the biotechnology also. So I'm curating for biotechnology also, right? So you'll get it maximum, I think within 2 days, you'll get it from my end. The rankers batch is ongoing. We have exam cracker modules, limited period offer till 8th of January. You can get these books for 1399, and that's about it. And you can join in the Lakshya batch as well. Every link of every segment is available in the comment section and in the chat section and in your description as well. So join in and prepare with us in the best manner. Okay, this is a Lakshya batch, completely free of cost, for you to uh, have a guidance, for you to have a day on like daily nudge for you to prepare in and perform in the best manner in the upcoming examinations. Okay, so bye everyone, take care. And right now we have more than almost 900, not more than, exactly 891 members are here, and they are preparing with us in the free manner in our classes. Okay, and the groups. So that's about it. Let's meet in the next class. Bye everyone, take care. Let's meet